All right, everyone. Today we are going over the Cellular Respiration Lab for germinating seeds. You have the document on your Google Classroom that you can follow along. So let's do an overview of our materials first. You should have a large beaker full of room temperature water, 2 small beakers, one that has something called manometer fluid that's red, and one that has potassium hydroxide. You should have 4 of these syringes that have a capillary tube on top. These are called respirometers. Be very careful when handling these because the capillary tubes are glass and quite fragile. You should also have a glass stirring rod, pipette, some cotton balls, a non-absorbent filler, a permanent marker, and some tongs. The experimental groups that we're using today are going to be these germinating seeds and these non-germinating seeds. You should have 10 of each. So to start, the first thing you will do is take one of your respirometers and we want to coat the inside of the capillary tube with our red manometer fluid. So I'm just going to withdraw a very small amount of the red fluid into my respirometer. And then I will pull the syringe down to coat the whole inside of the capillary tube with the red fluid. And then I will go ahead and eject that back out into my beaker. So now I've created an inside coating of this red fluid. Now the next thing you'll do is you will remove this plunger and get a cotton ball and split that in half. And you'll put half of a cotton ball inside of your respirometer and use the stirring rod to push that cotton down in there. Okay. The next step is to soak this cotton with our 15% potassium hydroxide solution. But now this potassium hydroxide solution is a strong irritant and you do not want it to touch your skin. So be very careful when handling it and wear your safety glasses when you are using the potassium hydroxide. We want 0.5 milliliters of this, which brings us halfway up the pipette. So I'll go ahead and take out around 0.5 milliliters and go ahead and carefully drip that onto my cotton ball, which will then absorb that potassium hydroxide solution. Now, we don't want this potassium hydroxide solution to touch our beans, our seeds here, because it, again, is a very caustic material. And so we need to put a protective layer between them. What we'll place between them is something called a non-absorbent filler, so it will not absorb that potassium hydroxide. I'll grab a small amount of this non-absorbent filler, and I'll go ahead and stuff that into the bottom of the respirometer like this. Again, use your stirring rod to help you pack that down. So now I've created a protective boundary layer between the potassium hydroxide and the seeds that I will now be putting inside. So the next step is to place 10 of our germinating seeds into our respirometer. So I'll take our germinating seeds here, put 10 of them in, and then I want to replace the plunger. But before you replace the plunger, be careful because when you push the plunger down, you're actually going to be expelling some of that potassium hydroxide. So when you put the plunger in, you want to point this away from you into one of the sinks as you push the plunger down. So I'll go ahead and insert the plunger and I'll move it over to a sink over here and I will push the plunger down just a little bit until I get to the 4 milliliter mark. So I've compressed it down to 4 milliliters. So this is my one completed respirometer, right? I'm going to repeat this process for all of my three remaining respirometers. Now, the reason that you have 4 respirometers is because you will use 2 of these for the room temperature experiment, and you will use 2 of these for the higher temperature 40-degree trial, which we will use this water bath for. So you should go ahead and now make your second respirometer, but you're going to repeat the same steps, except instead of putting the germinated seeds, you will put the non-germinating seeds in. All right. So again, you take your cotton, you put it in here. You take some potassium hydroxide, 0.5 milliliters. I will skip that step for now. Put some of this non-absorbent filler afterwards. And then I'll put my 10 non-germinating seeds in. And repeat, face this to the sink, compress the plunger until I get to the 4 milliliter mark. So now I have my 2 respirometers made, one with germinating seeds and one with non-germinating seeds. And you can just tell the difference by looking at the seeds themselves. You will place both of these into a beaker of room temperature water. They should be mostly submerged, right? You may top off some water just to get them as submerged as you can. And you want to leave these in the water for a few minutes, right? 3 to 5 minutes, allowing the inside to equilibrate. So it's going to match the temperature of the room temperature water. All right, so let's say I waited 3 to 5 minutes. Now, once all of this system has equalized, I'm then going to use a pipette, and I want to put my manometer fluid into the tips of each of these pipettes, in each of these glass cylinders here, of these capillary tubes. Right? And what we will be doing is, once we put the red fluid on each of these tips, we can then see the red fluid being sucked down the capillary tube. And the speed at which it gets pulled down will give us some information about the rate of respiration of the seeds that are germinating versus the seeds that are non-germinating. Now, if you put your red fluid on the capillary tubes and the fluid comes out, right, it doesn't get pulled in, that means you need to give it a little bit more time to equalize and repeat the trial again. If it continues to happen, you can call me over. There may be a leak in your respirometer. So I'll go ahead and give it a try. So I'll take a little bit of this manometer fluid out, and I will just put just a tiny drop on each capillary tube at the tip, like that. So you can see that the tips of each of these capillary tubes are red. Now, once you've placed your manometer fluid in here, you're going to take a permanent marker and just mark the point, the bottom of the manometer fluid. So the very bottom of my manometer fluid, I mark it with a little dash on each of these trials. Then you're going to time every minute for 10 minutes or until the fluid reaches the bottom. And so I start my timer, and after 1 minute, then I'll use my marker again, and I will again mark the bottom of the manometer fluid every minute. And we can use those increments late after the lab. You're going to measure those increments to give you some information about the rate at which these seeds are doing cellular respiration. All right, so I've gone ahead and waited the 10 minutes and allowed the manometer fluid to travel. Now, in the germinating seeds, the manometer fluid traveled quite far down, and I was able to make these increments every minute for how far the manometer fluid traveled. Now, since I have these marks on my capillary tube for every minute, I can now calculate the rate of this reaction. right? We have the distance traveled divided by the time that it took, right? So we have the distance divided by the 1 minute, right? And that will give you the rate of respiration of these germinating seeds. Now, hopefully you saw that the non-germinating seeds, the capillary, the manometer fluid did not travel down very far. Now, if there was any movement of the manometer fluid, you have to take that into consideration when you are calculating the distances on your experimental respirometer, because any changes to your control group were due to some external force, right? Maybe you pushed the syringes down or you moved your beaker, which caused a pressure differential, and the manometer fluid then moved. So you have to subtract the distance that this moved by the distance that your experimental value moved, because both of these would have been affected by one of those outside variables, and you need to correct for that. So you can remove your two respirometers, place them on a paper towel and dry them, and then measure the distances to get your rate. Now you're going to repeat this experiment, but this time you're going to be placing your two new experimental groups into a 40-degree water bath. So we will be seeing how increasing the temperature affects the rate of respiration. And again, one of these should have your germinating seeds, and one of these should have your non-germinating seeds. Once you've completely finished your experiment, you can remove these plungers. Please put the germinating seeds back into their wet Petri dishes, but everything else that's in the syringe can be thrown away, and the syringes can be rinsed out. Again, please be careful because there is potassium hydroxide residue in each of these syringes. All right, that's the lab. Good luck.